The genes in our cells are the blueprints of our life. They control all the processes in our body from hair color to blood sugar by directing cells which proteins to make, how much to make, when, and where. Inside every cell is a long strand of the chemical DNA. The sequence of DNA is a specific lineup of chemical base pairs along its strands. The part of DNA that determines what proteins to produce and when is called a gene. First established in 1985 by Sir Alec Jeffreys, DNA testing has become an increasingly popular method of human identification and its applications in research. The applications of DNA testing or DNA fingerprinting within forensic science laboratories is often what most people think of when they hear about the DNA. Using DNA to match blood, hair or saliva to criminals is one of the purposes of testing DNA. It is also frequently used for wildlife studies, paternity testing, body identification, and in studies pertaining to human dispersion. While most aspects of DNA are identical in samples from all human beings, concentrating on identifying patterns called microsatellites reveals qualities specific and unique to the specific individual. During the early stages of this science, a DNA test was performed using an analysis called restriction fragment length polymorphism. Because this process was extremely time-consuming and required a great deal of DNA, new methods like polymerase chain reaction and amplified fragment length polymorphism have been developed and employed. The benefits of DNA testing are so many. In 1987, Colin Pitchfork became the first criminal to be caught as a result of DNA testing. The information provided by DNA tests has also helped wrongfully incarcerated people to reclaim their freedom.